Good morning, everyone. I'm Todd Klaenhammer from the Department of Food, Bioprocessing, and Nutrition Sciences, and I love to eat bacteria. And the reason is that we've all been eating bacteria for upwards of 10,000 years in various kinds of fermented foods. And wine and cheese and beer, spirits, they make people happy. And they're full of bacteria. And so my motto is, eat bacteria, get cultured. Jeff Gordon and I organized a symposium at the National Academy of Sciences 2 years ago on the topic of microbes and health, and this has been published as a special PNAS symposia. And the fact is, ladies and gentlemen, that when you eat bacteria, fermented foods, or probiotic supplements, what happens is that you could be eating between 1 and 10 trillion bacteria per dose. These bacteria, being acid tolerant for fermented foods, can survive passage through the stomach, enter the small intestine where the local population might be 10,000 per gram. Think about it. You're overwhelming the small intestine with good guy bacteria that you can safely eat. Lower in the GI tract, 10 trillion per gram. And so over the past decade, genomic sciences on genomes of good bacteria that you can eat have identified a variety of different mechanisms that are considered to be probiotic. They are clearly colonization resistance, maintain barrier integrity and function in the GI tract, and adaptive immunomodulation. These are facts now being shown for bacteria that you eat at those levels that are safe. My point, bacteria through the GI tract interact with dendritic cells through cell surface molecules that are on the bacteria, and they communicate to the immune system. A well-established point, again, for safe bacteria that you eat in foods and probiotic supplements. In our research at NC State, we've looked at how these bacteria have surface molecules. So this bacteria has cell surface proteins and things called lipoteichoic acids out of a gram-positive cell wall. That molecule, lipoteichoic acid, is inherently inflammatory. And we use genetic technologies developed here through genomics and molecular biology to delete the gene that adds glycerol units to cell wall lipoteichoic acid. The end result is you change that bacteria from being marginally immunomodulatory to completely anti-inflammatory through the production of interleukin-10, which is an anti-inflammatory cytokine. The end result of that is phenomenal in two aspects. One is mice, normal mice, that's a normal cell integrity of the GI tract. When induced with colitis, it looks like this. It's been completely broken down, and this is the characteristic of inflammatory bowel disease. Pain, a broken down GI tract. If you feed the anti-inflammatory Lactobacillus acidophilus that all of you have been eating in yogurt since 1972, when we feed the anti-inflammatory organism, we can reverse the colitis. And in fact, that's a colitis score for diarrhea, for the colitis. This is the colitis score for the animal mouse, and thank you very much, David, for the mouse models because they're fantastic. And it shows a complete reversion. Second, if you look at just colitis scores, there's the colitis mouse, there's normal acidophilus. This is the mouse that's been fed the anti-inflammatory. If you go to the right part of the screen, this is a mouse model for colon cancer. Simply enough, the mouse on the top has colonic polyps throughout the intestine. It's been genetically engineered to develop colon cancer. If we feed the anti-inflammatory Lactobacillus, basically that mouse now has virtually eliminated the development of colonic polyps in that particular model. We're excited about this, and it shows, in fact, that you can make a difference by feeding different good probiotic lactic acid bacteria in disease models. Also, extended roles for these organisms, because you can eat them, they're safe. You can think about delivering a variety of different biotherapeutic molecules to humans or animals And in this case, we've used a model called a protective antigen for anthrax. And through a variety of mechanisms where we genetically engineer that vaccine with a dendritic cell targeting peptide that improves the binding of the antigen to the immune cells in the intestine, we've shown that you can get 100% protection of mice against challenge by anthrax through oral delivery of a bacteria producing this vaccine. Everybody else dies if they get all the controls or they get the bacteria not producing the vaccine. And so what we've learned in the past decade is that bacteria that you eat at high levels that are safe and they're gorgeous, they can impact the immune system via oral delivery and have an impact on our immune system and for vaccine delivery as well as potentially anti-inflammatory protection against diseases. So the future is exciting. Good guy bacteria is the next phase. Thank you for the opportunity today.